I'm Brian Malow, and I'm speaking with Margot Bezrutczyk. She's a postdoc at Lawrence Berkeley Lab. Thanks for taking the time to talk to me today, Margot.
Thank you.
So you're a postdoc in the Biosciences Directorate at the Joint Genome Institute. And well, what kind of scientist are you? Are you a geneticist or a genomicist or anything? Or what do you identify as?
I consider myself a plant biologist primarily.
And have you always been interested in plants? Did you know you were going to be a scientist from the beginning? Were you always, was it this sort of science if it was?
No, absolutely not. I thought plants were really boring and I was not interested in them at all when I was in college. As a child, I wanted to be everything from a paleontologist to an architect to a painter. And I went into college interested in neurobiology. So that's what my undergraduate and master's degree are in, actually. But I fell in love with plants. And so I decided to pursue a PhD in this.
How does one fall in love with plants over like the brain, neuroscience and stuff? Like, what was it about plants?
That's a great question, because a lot of people say that the brain and how it works is so beautiful, but I find plants beautiful in a more fundamental way. And I got started in it because I was working as a gardener, and I moved to Kenya to work on a small farming nonprofit. And I was really interested in the problems that people were having with crops, growing them in marginal environments. So in addition to just being like fascinating, really alien organisms, there's a lot of problems that we need to solve regarding plants and crop physiology right now.
Interesting. So I know you're from New York. I'd like you to quickly trace the path to your postdoc position in Berkeley. And was that Kenya? Was that part of your academic path?
Not really, no. I had actually quit the PhD program that I was in for neuroscience, which impressed nobody. And I just flew to Kenya and decided I wanted to do something completely different. So working with them wasn't part of any kind of academic track. My academic path was I went to NYU for undergrad. I went to UCSD for this PhD program that I ended up mastering out of, and then went to Kenya and joined the Carnegie Institution as a technician. That's at Stanford. I moved to Germany, to the University of Dusseldorf to do my PhD. And now I'm very happy to be back in California.
Excellent. And so what is it? You're a postdoc, you're at the Joint Genome Institute. What are you working on? And what are we going to hear about in the research slam?
So now I'm working on plant microbe interactions, and specifically I'm studying the relationship between legumes, so like plants that are related to soy, and the fungi that naturally live in the soil that provide them with nutrients like phosphorus. And it's really important. Sorry.
Yeah, I'm sorry I cut you off.
Oh, it's really important to understand this interaction, because all of the crops that we grow are very much dependent on the positive interactions between the fungi and the plant to provide it with nutrients. We pour a lot of chemical fertilizers on the soils in addition to this, but we're actually running out of some of those. For instance, we are running out of phosphorus to mine from the earth. And so if we can make the plants, fully dependent on the microorganisms instead, we solve a major problem in agriculture.
Maybe you could elaborate on this. So plants, roots, the plant itself isn't the whole story with obtaining its nutrients and stuff from the soil.
Oh, no, absolutely.
It's got some symbiotic help.
Yeah, exactly. None of the plants that we see growing in the wild or on farms are growing in isolation. There are millions and millions of microorganisms that we're not aware of that are helping them survive.
And how do you study that? It seems like such a hard, tiny thing to study. And I know a little bit because I saw your pre-video, so I knew a little bit about what you do. And I just thought we could, without you giving your whole talk, you could tell us a little more detail. It sounds interesting.
Right. Yeah, no, I have to say, going from studying just plants to studying plant microbe interactions has been really hard because they are so much more difficult to study. The way that I am approaching this right now is using single cell sequencing and spatial transcriptomics. And in English, basically, I'm studying the individual cells that make up these interactions and figuring out what molecules are in each of those cells. And so I'm getting information from the plant, and I'm also getting some information from the cells of the fungus.
And so besides this interesting interaction, like what's the big picture? I guess you mentioned that in the case of like some tools we have, like fertilizers like phosphorus, we might actually be running out of. What is the big picture here? It's like, okay, so you're studying this little tiny interaction there. What does it matter to us, the non-scientist?
Right. So we dump like metric tons of fertilizer on our soils every year. And in addition to this not being sustainable in terms of, being able to mine the fertilizers in the first place, nitrogen produces a lot of greenhouse gases when we produce it. And they actually all get washed out into the rivers and then into the Gulf of Mexico, creating dead zones. It's an ecological disaster in addition to being like fundamentally not sustainable. So it's a problem that needs to be solved. Like somebody needs to find a way to get nutrients into the plant's roots some other way. And at the moment, this is one of the most promising ways to do that.
I mean, didn't for millions of years plants have just a fine amount of nutrients? Is it because of our growing population, our need to be like efficient?
It's more like the crops that we grow right now, if we're going to be getting the yield from them that we are accustomed to getting, we need to provide them with this very high level of like supplemental nutrients.
Okay.
And I mean, yeah, like the truth is we could grow less corn. That's one of the major consumers of these nutrients. And most of the corn that we grow goes to corn biofuels, which is not particularly sustainable, and to feeding animals that we then consume. So if we could cut those things out, actually, yes, that would be another way to reduce this. But that's a societal issue, not a plant biology issue.
Yeah. So yeah, this is a big issue. What gets you motivated and out of bed to work on this? Because I'm sure like every day isn't always that exciting with the drudgery of actual day-to-day work when you're working on, even if you love it, there's a certain day-to-day kind of, what gets you up and motivates you to do this kind of work?
Well, I mean, I have to say, I do think that the problem of needing to fertilize plants using microorganisms is something that does need to be solved. So I'm very happy to be working on something that I think is critical for the future of agriculture. I also really like working with plants. And to be honest, single cell sequencing is such a hot area right now that when you get the data back, it is exciting. It's not really drudgery to analyze this data. I guess that's kind of a geeky answer, but
good. You like it.
Yeah, I actually like doing this.
And what about, so how are you feeling about doing the SLAM? Have you done something like this before, or have you done one of the research SLAMs?
I've never done a research SLAM before. I'm a little nervous because it is my first one, but mostly I see it as an opportunity to get a little bit better at science communication, which is something that I think is really important.
Yeah, could you elaborate on that? That was my follow-up is, what about that? Because your research is a full-time job. What do you think about the need or the role of public outreach for scientists?
I like to do this too. So I used to teach classes where we would have kids come in from elementary schools and middle schools, and I would teach them about plant biology. I would bring the plants in, and they're so engaged. It's so easy to get them excited about this kind of thing when they see the beauty of these organisms. I think it's really important for scientists to do this in general because the public has like a very poor understanding of what we do. And so if it's something that you like, you should definitely take the time to do it. We need to inspire the next generation scientists.
You just said that the public has a poor understanding of what you do. Is there something could you elaborate on that? What do you wish they did know?
So I guess, I mean, broader than like appreciating that plants are interesting living organisms, I would say a very timely issue is that people don't seem to have a good understanding about science as a constantly evolving body of knowledge. And this is an issue right now because of COVID, for example, that, you know, people hear one thing from early studies, and when that information changes because the virus evolves or further studies are done, they simply distrust scientists rather than understanding that, like, this is just the process. This is just a new thing we're learning about. So I think that that's something that I wish people had a better understanding of.
Oh yeah, that's a big one and very important. And it always has been, but it sure has been obviously important these days. So I want to thank you for taking the time to talk to me, and good luck in this research slam.
Thank you.